Okay, we're getting into the fun stuff. Contaminants. Okay, so can you name any contaminants that could be of concern in your food? Residue, uh, something added, something produced during processing. I know you know plenty. I know you're dying to tell me. Yes, Jacob. Huh? Allergens. Yes, some people are allergic to different proteins, and we have big eight allergens that are identified by FDA as causing more than 90% of allergies. Friday? Yeah, go ahead. Aflatoxin, which is one from mycotoxins in general. Yes, Evan? What's that? Oh, detergents that get into food? Yeah, yes, but we're not covering them today. Marissa? Foreign contaminants like glass or insects or rodent hair, that's extraneous matter. We'll cover that on Wednesday. Anybody else had a hand up? Lauren? Pesticides. Antibiotics. Did you have your hand up? Megan, did you have your hand? Yes, that is extraneous matter. We'll cover it next Wednesday. What can be produced during processing that can be toxic? Acrylamide, furans. Acrylamide and furans, they can be produced during processing. What can be added into food for adulteration? Huh? Bacteria? Huh. Oh, like yeah, but yeah, microorganisms, but we're not covering them in this class. But yeah, melamine, adulteration to increase the protein perception or content of food based on the nitrogen amount in melamine. So there are so many different contaminants and residues that are of concern, and they can occur at any point in the chain, the food chain basically, starting with the farm, animals on antibiotic, food produce and pesticides, aflatoxin or mycotoxins due to fungal growth, and then they could come during transportation, contamination during transportation. If they are like fungal or mold growth in the truck that can get into the food, and then production of mycotoxin can happen, for example. During processing, contamination, either adulteration purposes or during frying of food, you can generate acrylamide, or introduction of allergens. So at any basically step of the food chain, contamination can occur. For example, packaging material, like bisphenol A, for example, it can leach from plastic into the beverage of either a juice or water bottle, or another chemical from ink. There has been an outbreak of a chemical from ink that got into a cereal product. Yeah, we'll have so many other examples. So basically a lot of hazards have been reported, and this is just the period of 2003 to 2007, but there are, in the book chapter, there are newer numbers, but this is based on an older estimate of reports of hazards. So they are divided into different categories. For example, chemical hazards. These can go under like sulfites, for example, are under chemical presence of sulfites. Sometimes sulfites are naturally present, other times they are added as preservative and to prevent browning. So some people are sensitive to sulfites. Nitrites, for example, they're added to cure meat and as preservatives. Heavy metal, for example, these are chemical examples. Mycotoxin is by itself a whole category, and a lot of hazard reports are due to mycotoxin presence due to fungal growth. Microbiological, which we are not concerned about in this class, but it's also a lot of hazard reports are microbiological. Others, such as fraud, for example, melamine, biological hazards like urine, feces of animals, and hair of rodents. Labeling from the ink or from the ink. Other chemical hazards such as acrylamide, for example, developed during processing. Hygiene issues, defective packaging, and all of that. So the categories that we're going to talk about today are three main categories: pesticides, mycotoxins, and drug residues, antibiotics that is. And for these, there has been governmental tolerance levels. So the FDA and USDA have developed tolerance level above which there could be health hazards. So these has been established through experimentation with animal models to determine the level above which the food should be rejected. Another category is allergen. Chemical additives, these are, for example, adulteration added for a purpose of fraud. And then these two, for example, additives are added for preservation, and sulfite prevent browning, and here is the curing of meat, the nitrite and nitrate as well. So for the main ones that we're going to talk about, there's this table in chapter 33, table 1. It kind of summarizes, which is a good one to study. It summarizes quantitative methods versus semi-quantitative and qualitative, or what we call screening methods. So if we look at pesticides, for example, you have the multi-residue versus single residue. What do we mean by multi-residue methods and single residue methods? That means methods being developed to quantitate a number of pesticides in one analysis. Whereas single residue method is a method that has been optimized for a particular pesticide, for one pesticide. All the extraction methods and the analytical method has been focused to extract and analyze one particular pesticide. When it's used usually to generate that tolerance level, for example. So if we want to be very specific with our tolerance level, we really need to have a method that is very specific to that one residue. Whereas multi-residue methods, these are mostly when we are quantitating a number of pesticides that are similar in characteristics, similar in structural characteristics, so they are extracted under the same conditions and can be analyzed using the same column, whether it's GC or HPLC. So here for pesticides, it says that GC is mostly used. This is true for the most part, but recently they have developed pesticides that are more polar and less volatile. So with those pesticides, now HPLC methods are more common. So meaning of pesticides, about those in a minute. So thin layer chromatography and enzyme inhibition and immunoassays can be quick methods of analysis for screening. With mycotoxins, mostly HPLC, you can do GC, immunoassays, electrophoresis, and for semi-quantitative and qualitative TLC and immunoassays. For antibiotics, HPLC, GC, immunoassays, and a bunch of other qualitative methods we'll talk about. GMO, PCR for DNA and ELISA, and lateral flow strip we talked about earlier. Allergens, same, and we can also detect allergens with LCMS, Tandem MS specifically, and lateral flow strip as well. Sulfite, there are a number of them. Most common is Monnier-Williams. We'll talk about that method. This is a method to remember. This is the most common method for sulfite. And then nitrite, it's a colorimetric method, or we can do ion chromatography. Qualitative, we can do ion selective electrode or test strips for nitrites and nitrates. General consideration. So like with anything, your sample needs to be homogeneous, and the sample needs to be representative of the population. And this is very critical because sometimes many of these chemicals or constituents of concerns are randomly distributed. That means they are not necessarily located equally across the sample, so like mycotoxin, for example. So we really have to be very careful in how we take out samples that are representative for the population, and how many replication is very important. Otherwise, we're going to miss detection, and we're going to accept the products although they might be contaminated. So it's a very critical step. Sampling is very critical in this case. Now, choice of method, it really depends on how complex the food sample is. Do we really need to dry the sample? Is it high moisture? Is it high fat? So what do we really need to do in preparation to extract our contaminants of concerns? What are we looking at? What are the properties of the analyte? Are they volatile? Do we use GC? Are they polar? Non-volatile, probably easier to use HPLC. Can we develop, based on their properties, can we develop ELISA methods or an immunoassay for them? So basically, choice of the method, it really depends on what we know about the analyte. Feasibility, how tedious is the method? How many steps are involved in the method? Are we looking for qualitative or quantitative data? Can we go by with a simpler method just to see if we do have a pesticide or not in the sample? Then maybe only TLC is needed. Cost, how costly that method is. And again, are we looking for multi-residue, or do we want a method that targets a single residue to develop those tolerance levels? So the steps really are involving the analyses, goes from sampling to the sample preparation, and oftentimes you want to extract your components of interest. Depending on what they are, there are different solvents, either aqueous or organic solvents. And then cleanup. Oftentimes you use solid phase extraction, for example, instead of using two liquid phases for extraction and purification. And solid phase extraction, Gary covered that in GC, I believe, so you want to review that. Solid phase extraction is you basically use a cartridge that has a resin similar to a stationary phase that will capture your compounds of interest. So you run your sample through a cartridge or a filter, and it captures your compounds of interest based on their characteristics. And then you concentrate your samples. If there is a need to derivatize for GC analysis, you derivatize. Then you go ahead with your analysis, in this case chromatographic analysis for separation, and then identification either by a regular GC detector or an HPLC detector, but oftentimes for these compounds we use MS. Many of these compounds we use mass spectrometry and then reporting. So this is basically what this figure shows, but in here it just specify that during sample preparation you remove the edible from non-edible fractions. For example, you peel the orange. We don't care about the orange peel. You homogenize by chopping, grinding, blending to make sure that your sample is well homogenized before extraction. Sometimes you use solvent extraction. Sometimes you, after solvent extraction, you do filtration for purification. And then the cleanup. And this is what I said about either you do size exclusion, adsorptive, or affinity chromatography for more cleanup and concentration, or you do solid phase extraction or solid phase micro extraction for smaller samples, or samples that have your constituent at a very low concentration. So solid phase extraction, you have this abbreviation for QU is for quick, easy, cheap, efficient, rugged, and safe. That's what it stands for. You don't really need to know it, but that's what it is. It's describing how efficient you need your solid phase extraction to be. Or you can clean up using accelerated solvent extraction that we talked about as well in, I believe, the fat lipids lecture, where you pressurize your solvent so that the boiling point will be higher, so you're more efficient in utilizing the solvent to extract your constituents. Oftentimes your detection is with chromatography, your analysis, I mean, is with chromatography. Sometimes you need derivatization because if it's a GC method, and most of the time identification is by MS. Other common methods are immunoassays that are semi-quantitative for many of these constituents. So going into pesticides specifically, there are so many pesticides used, but every pesticide that is used has to be registered in order to be legally used. So that when it's registered, there are tolerance level. So they won't be registered unless tolerance level have been identified, their toxicity has been identified. So I think now there's about 1,300 registered pesticides. And they are analyzed, like I said, using multi-residue analytical methods or single residue methods. The single residue methods are often used just to determine for the level of tolerance. Semi-quantitative, thin layer chromatography and enzyme inhibition, because often your pesticides inhibit the enzymes in the pests. So they've designed assays to determine the presence of pesticide based on the ability to inhibit that particular enzyme. So GC is most commonly used for pesticide residue determination unless we know that the pesticides are polar and non-volatile, then HPLC is used. Choice of columns or detectors is based on the analyte. What's in the analyte? Do we have sulfur and phosphorus? Then probably flame photometer would be used. If it has halogens or sulfur and nitrogen, electron capture. If you have phosphorus or nitrogen, thermoionic. So really it depends on what type of pesticides we have. And then MS is often used, and if we have an MS, we don't worry about any of these detectors. This is an example of 5 organophosphate pesticides using a GC analysis with FPD, which is the flame photometer detector. Internal and external standards are used. Internal standards, as you know already, they're used to account for sample preparation error and volume injection error, whereas external standards are needed for quantification to plot standard curves. So HPLC is used for polar, non-volatile, non-thermally stable analytes. Again, choice of column is dependent on the type of the analyte. We can use UV detector, fluorescence, or mostly MS detection. MS detection helps in identification, especially when we have MS in tandem. So basically, when you have an MS in tandem, you can take a fragment and fractionate it even more. So here, for example, it's too crowded, there's a lot of ions, very hard to detect and decipher. But when you take a fraction of that and run it through another MS, you get a clearer spectrum that you can relate to a standard much easier than in this case. So oftentimes MS in tandem is used for pesticide detection. Immunoassays are used, and because pesticides are small compounds, you can use competitive ELISA, where the pesticide is, the entire pesticide is the epitope. You only have one epitope. You cannot do sandwich ELISA in this case. So you have your analyte, you have the labeled analyte, well, labeled pesticide that is the same as your analyte, labeled with an enzyme. So these compete for the antibody on, immobilized on your bead or plate. So thin layer chromatography screening method, and this is the one that we use to test the inhibition of enzymes. So in this case, what you do is you run your sample on your thin layer chromatography, and then you plate, and then you spray the plate with enzyme solution. Choline esterase, in this case, is the enzyme that the pesticide aims at inhibiting in the pest. So when you spray your column, or not column, plate, and then you add a substrate, then the color will be formed. And then you can see here that there are zones where you don't have color. This means there are pesticides here that inhibited the action of the enzyme. So when the substrate was added, no product was generated, and that's how they determine presence of pesticides that inhibit the choline esterase enzyme. So mycotoxins is the next category we're going to talk about. It's a secondary metabolite generated by fungus, such as Aspergillus flavus, for example. They are chemically diverse. That means there are so many different types of mycotoxins. The major ones are aflatoxin and ochratoxins. So aflatoxins are potent carcinogens and immunosuppressive agents. Ochratoxin is a nephrotoxin that attacks the nerves. There are limits of tolerance level, but for some aflatoxins, tolerance level is 0. So for example, aflatoxin B, tolerance level is 0. So it shouldn't be present. For others, there is a tolerance level that is less than 15 parts per billion. So methods of detection, HPLC, common one is reverse phase chromatography, reverse phase HPLC with UV or fluorescence detection. You can also use thin layer chromatography for qualitative, competitive ELISA, or immunoaffinity chromatography. Most common is competitive ELISA and reverse phase HPLC. Next is drug residue. So these are antibiotics. So they are administered to animals for either therapeutic or subtherapeutic purposes. Therapeutic, like if the animal is infected, obviously they treat them with antibiotics. And subtherapeutic, sometimes the animals are treated with antibiotics, but at a low level to reduce instances of infection, prevent them from getting infected, that is, and to enhance their rate of weight gain. But whenever the animal is ready to be slaughtered, for example, they have to remove the antibiotic treatment ahead of time so that the body is cleansed from antibiotics because tolerance level is 0. So in milk and meat, there shouldn't be any antibiotics in there because if we get exposed to antibiotics, we end up having bacterial resistance or sensitivity to antibiotics. So some of these antibiotics, people have allergenicity towards or sensitivity towards. So you don't want them anyway in your milk or in the meat because some people are allergic to antibiotics. Another reason why they shouldn't be in milk, for example, because presence of antibiotics will interfere in the formation of, in the fermentation basically, and then formation of acid and flavor during aging of cheese. So screening assay, there is the very common one, which is the disc diffusion method. Oh, turbidity is measuring just the turbidity based on measuring absorption. But the disc diffusion method, here you have a plate with an auger, and then if you see here, these little dots are discs that you put your sample in that you are suspecting that it has antibiotics in. And then you allow the plate for microorganisms to grow, and usually they use Bacillus stereothermophilus. And then if there is antibiotic, it will leach out of the disc and prevent microbial growth around that disc. So you would see zones of inhibition, you call them, where you don't see any growth. Charm test is an immunoassay based kind of similar to an immunoassay. It's not really immunoassay. What it is you have your milk sample that you suspect antibiotics to be present there. You introduce a radio-labeled drug that is similar drug, that means antibiotics that you suspect present in your sample, but it's radio-labeled. And then you introduce bacteria to your sample. So then there will be competition. So the radio-labeled drug and the antibiotics that you probably present in your sample will compete to bind to the bacteria. So if you have antibiotics present in your milk sample, you have less radiography reading. So you will have less reading, basically. If you don't have antibiotics in the sample, your labeled drug will bind to the surface of bacteria and you have higher measurement. So quantitative measurements, reverse phase, HPLC, and GCMS is another method. Allergens is another category, so proteins from different sources, the big 8, milk, eggs, fish, shellfish, tree nut, peanuts, wheat, and soybeans are the big 8 allergens. By FDA, they are responsible to more than 90% of the allergy outbreaks or cases. So the Food Allergen and Labeling and Consumer Protection Act applies to all food regulated by FDA, and it requires that all ingredients are listed by their common names, so people would know what their source are. So are they coming from milk? Are they coming from soybean? So that people with allergy can avoid that. Also during manufacturing, if there is any exposure to an allergen, it needs to be stated on the label. So for example, M&M that doesn't have peanut in it, it does say on the label processed in a plant with peanut, where they process peanut. The most recalls in packaged food is based due to allergens, presence of allergens. This is, yeah, so they are really a problem. So because allergens are proteins, so they are measured using protein methods. So we can do ELISA, Western blot for a common qualitative method, or biosensor. That means we have antibodies immobilized on biosensors. But here I have an image. The reason I have this image is to illustrate that the way we extract the protein for analysis is very important. So if I suspect that a food contains an allergen, but it depends on how I'm extracting the protein, I may or may not detect that allergen. So in this example here, these are 2D gels, A and B, that were run, and then Western blot were run. You can see here there's both coming from peanut. You can see where the circles are. So the protein has been extracted with salt in the buffer, whereas B, the protein has been extracted without salt in the buffer. So without salt in the buffer, some of these proteins were not detected, were not present as allergens, whereas when salt was present, these proteins were found and detected as allergen using the Western blot technique. So the one consideration is the extraction methods for protein analysis. Another consideration for protein analysis is that when you process the protein, when you process the food, protein changes in its structure. So if you have set assays, immunoassays, and then the protein, because of processing, it changed in structure, it might not be detected by your assay, but it would still be present. So if a person consumes it, it's still going to cause allergenic reaction. So that is another disadvantage. DNA methods, so if the DNA from a particular source is present, you can detect that with PCR. But the disadvantage of that, if you're using in your food product an isolate, a protein isolate, and you're putting it in a bar, the protein isolate most likely will not contain a DNA material. So when you don't find the DNA material, that doesn't mean you don't have the allergen. So each method has really its advantages and disadvantages. So oftentimes it's good if we really want to be very sure to do a complementary set of analyses. Well, the list is long for contaminants of concern. So other than pesticides and drugs and mycotoxins and allergens, we have a whole list of contaminants and residue of concerns. So sulfite, for example, so there are legal limits for sulfite. There are legal limits for benzene, and benzene can form in a beverage that contains ascorbic acid and benzoate salt. So it can form, but there are legal limits for that. Furans and acrylamide can form during processing. Sulfites can be added or naturally present, like in wine, you have sulfite. Nitrites and nitrates are added for preservation and for curing meat. Melamine is an adulterant, and the rest we'll talk about them as well. So, and these are the categories. Some are banned completely, some have legal limits, Some are international contaminants like melamine, and some are approved for use but of concern like monosodium glutamate, the umami flavor. And some are natural constituents of concern. That means they occur due to processing of natural products. So potato, for example, in the presence of a reducing sugar and asparagine in frying oil means high temperature, you're going to form acrylamide. Furans are often formed in high sugar, high carbohydrate under high heat. Many of these contaminants, they are, the methods of analysis are really tedious. That means time consuming and expensive, and mostly you're using chromatography and mass spectrometry for detection. And the sample preparation cleanup is really lengthy. The problem is with the limit of detection is a major concern because some of these are really toxic at very, very low concentration. So we really want to detect them at very, very low levels. So oftentimes the methods are really tedious because you want to make sure that you follow every precaution and concentrate so that you can actually detect those those contaminants in your sample. There's always an effort ongoing to develop more rapid and inexpensive methods. So still work in progress. If we look at sulfites, so sulfites include sulfur or sulfiding agents, include sulfur dioxide, sulfurous acid, various inorganic sulfite salts. They can occur naturally or added to food to prevent as a preservative and to prevent browning. Some people have sensitivity to it, so they have kind of a reaction, not allergenic reaction, but kind of a asthma attack reaction to it. So the label should state if it contains sulfur, it should state that it contains sulfide if the concentration is greater than 10 parts per million. Most methods of analysis only detect a free form of sulfide plus some bound form. Not one method can determine all forms of sulfide in food. Sometimes you have to do a combination of methods. So, but the most common method is the Monnier-Williams method, which measures total sulfur dioxide. So, which measures the free sulfide plus a reproducible portion of bound sulfides. It is the method that the FDA recommends for labeling. And the way it's done is basically your sample is heated with HCl. So all of the sulfide, most of the sulfides are converted to SO2. And then the SO2, in the presence of nitrogen gas and H2O2, that's peroxide solution, it oxidizes SO2 into H2SO4. So the peroxide oxidizes SO2, and then you get H2SO4, which either is measured gravimetrically or by measuring the turbidity. That is the most common method, especially for labeling. There are different methods. The Ripper method, which is a titration method, and then an enzymatic method, and also an HPLC method. So, but the one that you really need to know is the Monnier-Williams. Sodium nitrite and nitrate, so these are preservatives, so they're added to cure meat, giving you that pinkish reddish color, and it prevents browning. And the way it functions is sodium nitrite in the presence of salt prevent the growth of Clostridium botulinum, for example. Sodium nitrate, the method, sodium nitrate can be converted readily to sodium nitrite. And sodium nitrate is naturally present in vegetables, some vegetables. of course, there are legal limits for them in meat and in water. And then the health concerns is sometimes residual nitrite in presence of heat. It forms nitrosamines, and these are carcinogens. High levels of these compounds can cause a blood kind of disease, which is methemoglobinemia, type of anemia. So the AAC methods, it's basically colorimetric method and ion chromatography method. So the colorimetric method or xylanol method, here the sample is placed with sulfuric acid and treated with xylanol to produce 6-nitroxylanol, which is distilled into water, isopropanol, alcohol, ammonia, hydroxide mixture. The ammonium salt provides a yellow color, and then you measure absorption. And then you can use ion chromatography to separate the nitrites and nitrates, and then use conductivity detector to measure their presence. So there are ion selective electrodes similar to those that we described for mineral analysis chapter, otherwise test strips. Can be used, like the AquaCheck nitrate test strip. And yeah, you just don't worry about the detail. Just know ion selective electrodes and test strips for those particular rapid methods. So this table is present in your chapter, and it's a very good table to actually study from. Because if I ask you a question similar to the review session today, I gave you a food product and asked you to identify a compound that can be present in it. So ACrylamide, it is formed in carbohydrate-rich food cooked at high temperature. So why is it a concern? It's a carcinogen. What kind of food it can be present? Fried food, high in carbohydrate. And what method of analysis? LCMS MS. So this is the same, similar is for the rest of them. So it is a good, if nothing else, study this table from this chapter. So melamine adulteration, we mentioned before that it has been used as a fraud to increase the detection of protein based on nitrogen analysis. So melamine has so many amine groups in it, obviously, and nitrogen. So if we're doing DUMAS or we're doing KELDA, we're going to detect this nitrogen as protein. So melamine is added to match a certain required amount of protein in the product. So it was, firstly it was detected in pet food, and then it was detected in infant formulas. Originally it passed detection because the method that were used to detect protein is based on nitrogen, so it was not detected. But when sickness or illnesses were reported, then these food products, they were analyzed further for adulterants, and they discovered the presence of melamine. So it causes high blood pressure, blood in the urine, kidney infection, and it can be fatal. So methods, LCMS Tandem MS, that has a limit of detection of 0.25 micrograms per gram. And GCMS, limit of detection is 10 micrograms per gram. Other methods, electrospray ionization MS, that is direct, you don't have to do a chromatographic separation. Near infrared immunoassays. time. Thank you. I wasn't looking at the time.